Welcome everybody to our continuing coverage of ASGCT 2022 here in Washington, D.C. I'm Greg Slobodkin, Editor-in-Chief of ScienceBoard.net, and I am glad to have with me Thomas McCauley, Chief Scientific Officer for Omega Therapeutics. Welcome, Thomas.
Thank you, Greg. Thanks for the opportunity to talk about Omega today.
It's fascinating to talk about this topic, and we were talking a little bit before we started this. Can you tell us a little bit about Omega Therapeutics, when it was founded, and what its mission is?
Sure. So Omega was founded out of Flagship Pioneering in 2017. And Omega is really pioneering a deterministic therapeutic development platform that harnesses the power of epigenetics, which is really nature's operating system for controlling gene expression and cell differentiation. And we leverage our foundational understanding of how epigenetics controls gene expression, and particularly the role of these insulated genomic domains, or IGDs as we call them, as fundamental units of that control to enable us to very rapidly design programmable mRNA therapeutics to control gene expression to treat and potentially cure really any disease.
You know, it's interesting. I heard your CEO talk about, tapping into this universal operating system in nature and then being able to control gene expression up or down. I mean, that is essentially the essence of what you're after, right?
It absolutely is, in the sense that epigenetic mechanisms are nature's operating system for controlling our genome. So while the sequence of all of our genome is the same in all of our cells, those function differentially, obviously, to create sort of a nerve cell versus a heart cell versus a brain cell or hair cell or something. And so nature has organized our genes and their regulatory elements into these three-dimensional loops of chromatin that we call insulated genomic domains that are bounded at their base and effectively insulated. So it allows the genome to, through these epigenomic marks that form the program for each cell, to determine which sets of genes are going to be turned on or off at any given moment throughout the life of an organism, and to allow cells to take that same complement of genes and turn sets of them on or off independently without affecting the genes nearby. And so we're simply taking a page from nature's operating system, as you said, and co-opting that for therapeutic purposes and designing molecules, in this case, delivered as mRNA, that allow us to utilize those exact same tools to affect gene control.
And what diseases are you specifically targeting?
So we have as you might imagine, this is a very broadly based platform that we believe has applicability in oncology, where our lead program is, for example, is sort of a poster child for epigenetic dysregulation and therefore epigenetic therapy. But really, to the extent that, again, epigenetics controls every aspect of gene expression sort of throughout life, we believe this is applicable across the board, really, certainly in monogenic diseases, but also importantly in complex multigenic diseases. And honestly, most real-world diseases are driven by many different genes. And so because multiple genes are typically organized into these IGDs, it offers us a very convenient, very elegant physical handle by which to manipulate expression of multiple genes, which often control whole disease processes or biological pathways, potentially with a single therapeutic. So it's really a powerful paradigm.
And here at ASGCT, you came up with some preclinical data, right?
That's correct. So we were presenting data here following on the presentation we did at AACR just a little while ago on our hepatocellular carcinoma program. Here we were presenting data on our non-small cell lung cancer program. So here we're targeting an IGD, in this case, that contains the MYC gene, which is, as you probably know, is implicated in essentially 50% of cancers, essentially all metastatic cancers. It's right. a key driver in a number of different conditions and essential for cell function. And so, we have, through our study, understood that there are, significant portion of the non-small cell population we believe is driven by overexpression of MYC. And so we've designed a particular epigenomic controller here to target that in the context of non-small lung cancer specifically. And we're showing data that, as designed, our OECs are delivering very, very, precise, specific epigenomic reprogramming of the IGD itself, and that's driving the downstream changes you would expect in terms of modulating MYC expression. So certainly we're driving down expression at the mRNA and the protein level, but also MYC controls probably 15 or 20% of the genome downstream. Right. And so you see things like proliferation markers go down, and you see markers of apoptosis and things go up. And so the net result of that is that you see cell killing in tumor cells, which are overly dependent on MYC because many of the signaling cascades in different types of cancer actually converge onto the MYC pathway, which is downstream of many of them. So it's a unique non-redundant pinch point to really target for many different kinds of cancer.
Right.
And we're able to show in vitro that we see very, very strong tumor killing, both as a single agent, but also in combination with many of sort of MEK and EGFR inhibitors that are used as part of the standard of care. And we believe that this will also be additive or synergistic with other agents, like IO agents, for example. We've also shown, most importantly from a safety perspective, that while we target overly MYC-dependent cancer cells, we don't have that same effect on healthy lung cells. So those are spared. And then most importantly, on the efficacy side, in terms of in vivo data, we showed very significant tumor inhibition against single agent in a number of non-small cell lung models.
Wow, that's impressive. In terms of diseases, because you said that there is wide application here for your technology, is there an effort though to go after unmet needs, in terms of disease?
Absolutely. I mean, we are, a broad-based platform approach, but, we approach diseases the way that most drug developers do, which is to look where the unmet need is. I mean, I think the sort of genesis and long running conferences like this that are built around the tools and the modalities that are used to treat the genetic bases of all these diseases speak to the fact that there's still a tremendous amount of unmet need, and not just in people I think often associate genetic therapies, for example, with rare monogenic diseases, but really most diseases have a genetic dysregulation at their core. And so, the therapies that others and we particularly are developing really, we believe, are applicable to all of those in the full breadth.
Yeah. Can we talk about your pipeline? Because there's, and I want to get this correct here, there is an investigational drug that you're applying for, I think this year, right? The first half of this year.
That's correct. So we are on track to file an investigational new drug application in the first half of this year. So coming up rapidly on that for the hepatocellular carcinoma program, as I mentioned, where we hope to be in the clinic for that sort of before the end of this year.
Oh, wow.
And then following on very closely from that, we've said before that we were going to nominate 2 additional development candidate programs as well. We've not specified what those are, but
okay. Do you want to give us a hint today?
Well, certainly the non-small cell program is among the ones in contention. I mean, part of our fundamental portfolio strategy as a platform-based company is to have multiple options in terms of what we put into the clinic, and mitigating our risk as a company in terms of differentiating amongst targets and target cell types and that type of thing. So. I think from a biology perspective, the risk in the MYC indication is low.
Right.
But we're continually looking, and I should note too, as you look across our pipeline on our website, for example, you can see we're focused in regenerative medicine, oncology, immunology, and other kind of complex multigenic diseases, and then monogenic diseases as well. In part because we are looking to sort of stake out the 4 corners of the platform in terms of the real breadth of applicability, but also to mitigate risk across targets potentially. But one thing I would note is all of the targets that we've publicly disclosed, certainly, and others, are ones where we believe that sort of causal link in terms of the disease biology between the dysregulation of the target gene or genes, and the disease phenotype is well established.
Yeah. You talked about IGDs, and what within that is an epigenomic programming platform, right? I mean, is that sort of the combination there of the 2 technologies?
Yeah, so the pillars of the platform, if you want to think of it that way, the first is sort of the central role of IGDs in controlling gene expression and our understanding of how that functions. One of those key pillars is the sort of computational and functional genomic expertise that we apply to interrogate an IGD initially in silico to understand where the genes inside it sit relative to the regulatory elements and the loop anchors, and how it functions in a healthy state, and then how that goes awry in a disease state. And then that very rapidly leads us, again, in a sort of rational design approach that's computationally guided, to the design of a programmable mRNA medicine that allows us to target a very specific intervention point. We call them epizips, sequence intervention points, around the IGD, which could be a CTCF molecule, it could be an enhancer element, it could be the gene itself. Oftentimes, synergistic combinations of those, to tune gene expression to the level that's necessary to rescue that disease phenotype. And that's an important point, I would say, for the platform, is that we're co-opting nature's operating system for controlling, modulating gene expression. And the idea is not to do things in a binary way to sort of turn things either all the way on or all the way off, but to really tune them only as much up or down as necessary, again, to alleviate that disease phenotype. And the other key piece of this that I would mention, again, taking a page from nature, is that we're doing this without cutting the genome. We're not making single or double-stranded breaks in the DNA. We're not inserting anything into it. And so inherently, in that respect, we believe that this should be not only potent, but also significantly safer than other approaches which rely on editing the genome or inserting things into it.
Yeah, I mean, it's interesting. You and I were speaking previously about the FDA guidance that came out recently.
That's correct.
And how that has interesting ramifications for, this kind of therapy down the road.
Yeah, absolutely. And so, the editing approaches, whether it's sort of editing a cell, from a cell therapy perspective or, directly editing the genome itself, comes with some liability from relying primarily on, endogenous DNA repair mechanisms to sort of sew up those breaks. And so to the extent that many of the modalities out there are creating, constitutive machinery that can cause some of those breaks over time, oncogenic risk and risk of other untoward things, goes up significantly. And so the FDA has recognized that in, mandating a really significant 15-year follow-up in terms of clinical, for the clinical populations for those indications. I mean, certainly I think it's warranted, but part of our conscious decision going after an epigenetic mechanism where, again, we can design specificity, as we talked about, tunability in terms of amount, degree of expression, but also durability of effect. So we can tune these to and engineer these to last anywhere from a couple of days to a couple of weeks to a couple of months in terms of their effect. But the actual molecule itself, the mRNA, and then the protein that it encodes, both the binding domain and the epigenetic effector, are degraded over the course of a couple of days. So you've effectively separated the pharmacokinetics from the pharmacodynamics of this modality. And so you can have very long-lived pharmacology and effect without the liability of having the drug on board for that whole time, and certainly without making any changes to the genetic sequence.
Yeah, wow, these are exciting times for precision genomics. I mean, it's just amazing.
They really are. It's amazing to see, at the conference, over the past week in particular, but just over the last 20 years, the advances. And I think, obviously we're focused on our own development, but I think that the broader picture is that there are a lot more therapeutic options now for patients living with diseases with genetic basis than there ever were. So that's exciting to me as a drug developer.
Absolutely, absolutely. Well, thank you for your time, Thomas. I really appreciate it. And
you're very welcome.
And stick with us. We'll have more coverage from ASGCT. Thanks.